Okay, John, go ahead.
Oh, so basically, I'm going to miss the chance to interact with the audience as I do in a normal WABA presentation, which is always one of the highlights of being able to present. Great questions, great interaction, feedback, but hey, these are the times that we're at. So the presentation tonight is going to be intermediate. Okay, so intermediate means you're stepping up, ladies and gentlemen. It's not basic butterfly biology anymore. We're getting to some nitty gritty kind of stuff. And some of that's going to be, a little bit hard to go. So I talked Paulette into letting me actually read the text so that I can put the proper emphasis where it belongs. At any rate, tonight we're going to be discussing anatomy, phylogeny, and a little history. We'll do a little history first, all right? One of the things I wanted to talk about is, like, why are people doing research on butterflies? Well, the popularity of butterflies actually goes a long ways. It is a, how do I get rid of this thing? Oh, never mind. I grew up in 1961, my first butterfly book, 1962, actually. I got my first butterfly book in 1961, published then. And, like, it was like the only thing around, all right? So I'm going to talk a little bit about that. And then, some of the results have come from, the expansion of butterfly awareness. And then at the end, we'll talk about some uncomfortable things, butterfly name changes and such. So, like, what I did was I just really quickly, and if you guys really want to have, a selection of all the books, I have this all documented, but I'm going to slide through it pretty quick. Okay, so these are the classic works of the 19th century. They're amazing. I think that everyone should look at Samuel Hubbard Scudder, the best butterfly book in 3 volumes ever produced for our nation. And it's just outdated, but perusing it, it's wonderful. So we're gonna run through this 1905, 1931, that was my second butterfly book, 1951, 1961, 1975. And it was like, that was kind of the point where things started to turn, because after that, this guy named Pyle, you guys know about that guy? I think he's online. He began a series of books that ended up with Pyle in 2018. So what's really clear is that this increase in butterfly books reflects an increase in butterfly awareness or interest in butterflies. And as a result, it includes, all of us like you and I tonight, and then serious researchers who grew up as kids reading all these books. Now, I always tell this, it's like butterfly pursuit it's very, very serious business, and it can consume you. I am consumed. Once infected or afflicted, you may have that fever for life. And my buddy Kiyoshi Haruma quite adequately described it as crazy for butterflies. That's actually a translation from Japanese. I think it's kakacho, but I'm not sure, right? So what I'm going to do is actually look at some of this amazing work that has been done in basic 2 examples, but this one's going to be main stage because this is a revolutionary publication. And I highlighted Gary Bernard's name. If we were lucky, he'd be watching tonight, and I could give him the praise that he's due, but he's one of ours. He's presented to WABA a number of occasions, and he comes to meetings sometimes just so he can, watch us. But I want to read the title so that we can kind of get through this whole thing intelligibly, because it's going to get worse than that. Physical and behavioral adaptations to prevent overheating of the living wings of butterflies. Now, when I read that title, I didn't actually grasp what they were saying. They were saying exactly what the title says, and it is exactly living wings. So I will just move on and start to look at some of this, in the context of basic butterfly biology. So, we go back, to, some of the original contentions in basic butterfly biology. One of them is, how you wake and warm. This is the standard model, and it's not in error. These are mechanically still correct, but this was the passive thermoregulation, basking. We all have seen butterflies dorsally basking like that Lorquin's admiral there. We've seen lateral basking like that wood nymph there. And of course, you can turn toward the sun that's cooling, that's all of these things are still in play. But we did this with the under the misimpression that wings were passive, like solar panels, or maybe not appropriately, just like solar panels with silicon chips, right? So this is another representation of the same thing. You see that arrow coming in from the sun, and then you see the radiation that takes place from the wings and to the substrate, wings to open air to the substrate, modified by whether or not a wing is open or closed, or what direction the butterfly is oriented. That's not really changed, except, okay, so I'm going to go with excerpts and I'm going to read them. The wings of Lepidoptera contain a matrix of living cells whose function requires appropriate temperatures. And I was like, they keep talking about living cells. I mean, I'm reading this for the first time when this is all happening. Here we analyze butterfly wings across a wide range of simulated environmental conditions and find that regions containing living cells are maintained at cooler temperatures. There it is again, all right? Diverse scale nanostructures. Okay, now we're talking about nanostructures. We're going to look at those in a minute, but diverse scale nanostructures and non-uniform cuticle and thicknesses create a heterogeneous distribution of radiative cooling that selectively reduces the temperature of structures such as endocorneal. Wing veins and endocorneal organs. They're going to come up later. Now, I did not construct this text. This is from the abstract. And about this time, I'm starting to realize, whoa. So these are nanostructures, okay, with some other things. You can see the highly developed complex structures that are part of the butterfly wing. And you can see there's, open air spaces, and there's, certainly a lot of external structure to that. And it's like, that's what they're talking about. But I'm going to go back to look at this just so that you can see that these different structures in the wing are aiding radiative cooling, selectively reducing the temperature. It's like, all right, so we're onto something. These tissues are supplied by circulatory, neural, and tracheal systems throughout the adult lifetime. Indicating that the insect wing is a dynamic living structure. About here, I'm starting to lose, contact. It's like, wait a minute, is this a conspiracy theory? Is QAnon involved? So in both Satyrium carivorus and Parhasius M. album, we observed a wing heart that beats endogenously, that is originating internally to the wing, at a rate of a few dozen times per minute, and appears to draw hemolymph unidirectionally through the scent pad. It's like, really? Okay, so I'm going to take a look at that. This is actually a picture, a photograph of a scent pad. Now it's been, it's obviously, been manufactured to show, I don't know, can you see the wing heart there? All of a sudden I noticed that in my, oh yeah, there it is. Okay. This is a scent pad on the wing through which the veins are flowing, and through which the membranes, is showing the pumping action. And basically the heart is pumping and sucking, the material through that scent pad. This is what they're talking about when they say wing heart. And I'm like, by this time I'm flabbergasted. I'm reading this for the first time and I am flabbergasted. I went back and looked at my notes just to make sure how flabbergasted I was. I was very flabbergasted. They go on, and again, I can do no better than to quote from them because they are actually exquisite. I cheated. I saw that word on the page. Behavioral assays show that butterflies use wings to sense visible and infrared radiation, responding with specialized behaviors, we just looked at those, to prevent overheating of their wings. Our work highlights the physiological importance of wing temperature and how it is exquisitely regulated by structural and behavioral adaptations, which is like, yeah, they're talking about the same thing. We investigate the thermodynamics and thermoregulatory properties of the wing itself, first by observing the distribution and persistence over the entire adult lifespan of living tissues in the wings of butterflies. And then by studying the thermodynamics, it sounds like they're making a nuclear bomb here, you know, of the wing and the physical and behavioral adaptations that modulate wing temperatures in ways that protect these tissues. All right. We develop an infrared hyperspectral multi-wavelength imaging technique that permits accurate temperature mapping of objects that are both lightweight and translucent, like butterfly wings, in the mid-infrared. We find that wings are extremely heterogeneous in their thermodynamic properties, with wing veins and other living parts of the wing being cooler than intervein regions under solar radiation. Now, these are dead butterflies, prepared butterflies, and these are the imaging techniques. So they're showing that even in dead butterflies, the cooler regions are recognizable. All right. So the idea is to show that the technology, and here's some more. You can kind of recognize, what the butterflies look like without the infrared. But I wanted to show that, this is a technique that's highly evolved. I suspect that Gary Bernard had a great deal to do with this. I'm just guessing because that's the kind of guy he is. He said, "I'm not a scientist, I'm an engineer." Scientific engineer at least. And then these are what the living butterflies look like with that camera. It shows quite clearly that there is some, control going on of the wing surfaces. And, it shows that this is an active process in living butterflies. You can kind of see, the text is not really intended to be read. If you can, great. It does show that there's a laser effect and they are showing the direction. If you, want to read the paper, it's available downline. I'll make all of that, available to you, but just wanted to get to the good stuff. Okay. So this is actually a living butterfly and the representation of it in the infrared instrumentation. It's like, wow, really, really great stuff. Okay, one last thing here before I get carried away. We observed that despite the wings' wide variation in visible coloration and pattern, which we all know butterflies have, the average temperature of the scent patches, pads, and wing veins that contain living cells, including hemolymph cells, androconial cells, and mechanical and thermal sensilla, which are nerve endings, is always lower than that of the remaining non-living parts of the wings, regions with the fused membranes. So I'm like, at this point, excuse me, oh no, this isn't at all what I learned. We were taught that after eclosion, butterflies' wings dried, and they, from that point thenceforward, were instruments of, movement and motivation, and they basically were dead, incapable of only passive activity, through regulatory activity. Certainly, apparently, this is not so. So you got a time here. I said I put a pause in here. Paulette, is that about right for you?
Yeah, that's fine. Can you go back to some of those thermal images and show which are the cooler parts? I mean, there's different colors, but it's hard to, unless you know what the key is, to understand which are the cooler parts and which are the warmer parts.
Well, that's a good question. I'm not sure that I can access the bright parts. Okay, like if you look at this image here and on the right, you see the butterflies is represented by some really bright parts. Those are probably warm.
Yeah, I see the scale over here now, next to the Eumaeus Atala, to the left of that.
Oh, there you go. Yeah,
I see it
You
now.
said that very well. Eumaeus Atala. That's nice.
May I ask what an androconial cell is?
Okay, now that's good. We're going to get to that. It actually is a specialized sex cell that is present in males only, and it is to elicit a mating response from the proper female. So it's very important in mating, and we're going to enhance that. That's basically one of the major upshots of this paper. It's usually not like a wing scale, except in basic structure. It's attached more or less the same way the wing scale is, but we'll get to that and we'll see it. But they're specialized scales, so like, yeah, that's good. I forgot that everyone knows butterfly lingo. Endocorneal scales can be spread all over the wing and often are in some species, or can be concentrated in certain parts of the wing, which we will see, or they can be actually tucked into little pockets, which we will also see. But that's a great question, and you're absolutely right. I should have been a little bit cleverer. Any more?
Yeah, there's a question from David Droppers about what is the advantage of having wings with living tissue versus dead structures. He suggests that having dead structures would be advantageous in that they would require less to maintain, as well as being less prone to overheating.
Well, here's the thing. You can ask that question. It's a legitimate question, but the interesting thing about it is these are active. The wing heart, for instance, is an active process. So it's moving warmer to cooler to warmer to cooler to warmer to cooler as a part of an active circulation system, which means it's much more efficient. So you can have a passive thermoregulatory device be very effective. We always thought it was sufficient, but it could be much more efficient. And when you think about butterflies, they don't really have time, to spend taking a long time to warm up or to cool down. I mean, warming up is probably more critical because, when a butterfly becomes active, it has to enter that predator avoidance phase rather quickly. And so this will definitely increase the efficiency of all of the aspects of the wing. Now we'll get into beyond the thermoregulatory function, which is really, really important. But I think, for me, it's amazing what a living wing means to butterflies. And, so like, if we got some more questions, we'll go for them. Otherwise, I want to move on.
Okay, I just want to let you know that Gary Bernard is joined us, and so we can either hear his thoughts now or we can wait until the end of your formal presentation, John.
Okay, well, I'm glad that he joined. Hey, Gary, you see that? I got it all highlighted, and that's your name there, and I made sure everybody knows that you're our hero. All right?
Okay, Gary suggests that we wait until the end of the talk to engage more. So
Okay.
You're free to go on, John.
All right, I am free. It sounds like a Who song. Okay, so reproductive and mating behavior, mate selection, rejection. We have to kind of review this a little bit. It's no doubt the most important thing that all organisms must do is stay alive. And the reason why it's important for them to stay alive because reproduction requires it. So the second most important thing that organisms do is get mated. This is a process that's facilitated by, clustering, for mate locations. The people that look for butterflies will go to hilltops because butterflies congregate on hilltops. Virgin females will too. That's not the only place they do it. That's just one style. And then the perching behavior, finding a female, determining that's a female, determining that she's available. All of this is really important because it's a mate choice. It has to be pretty close to 100%. If butterflies mate irresponsibly, then that's going to be deleterious to the population. Of course, it gets selected out real quick. So, how do you do this? Well, there's ways that you can do this. And one of them is these sex scales. Androconial patch. I told you we get there. Here we are. These androconial patches are collections of specialized scales that actually enable mate recognition. And since males, they're basically just trying to court, so they're not real, it's not a real important thing. Their decision-making process in the matter isn't as important as the female decision-making process. So, in this particular butterfly, which is the great Arctic, males have this very prominent, sex patch in the forewing dorsal. Females lack. It's like, whoa, they almost look like different butterflies. And I can say, knowing these butterflies very well, that females, will quickly reject anything that's not a male, and often reject many males if they're already mated. And this is why, because they can smell. Now I say smell, it's not smell, but it's some kind of chemistry. I don't know, that's another whole program, but at least, we know that these sex patches play a big role in mate recognition. And we all know the monog, that pestiferous insect that eats our milkweeds, a vagrant pest of our milkweeds, is notably recognizable by male sex patches. You can see that right there, that sex patch, and you can see the difference between males and females. It's not as striking as in the great Arctic, but it's important to recognize that that is the same thing we're talking about. You can see it's more confined. And so that's what I was saying earlier, is that sex scales androconia may be widely expressed over the wing surface, or they may be extremely localized and specialized places. So here's where we get a chance to look at some of these androconia and a chance to see how they are expressed in different kinds of butterflies. In the upper right at 110 is a typical Satirid location for androconial patches. They look like they're separated into different cells, and they may be sometimes separated, but a lot of times they're actually in a single cell, like in 132, expressed between the discal cell and the adjacent cell. But the scales themselves are really interesting. They can be very different from normal wing scale, long and skinny with a little little tendrils at the end. Now, if you think about it, why would a scale have tendrils at the end? Well, better to dissipate ephemeral chemicals, right? And that's not an unusual feature. As you look around, you'll see that there's a lot of these hairy things. Well, that's because the chemicals are going up the length of that scale, and they're getting into the atmosphere and dispensed more rapidly. You got to think about a mating butterfly, courting butterfly, is you don't want to make its case really quickly. So it only has a little bit of time before the female will reject him or him, and therefore having scales that really facilitate the dispensation of these chemicals is an important function. Now, the other thing you might see is the occurrence of sex scales along veins. And we have a couple of Washington butterflies that will show that. I will actually show them in a minute. But I wanted to make sure that everyone saw the variety of sex scales and where they can be on a wing. And this is, slide one of another one later. But here's a European frit, notoriously, margined with these sex scales along the veins in the forewing. You see how thick those veins are? That's sex scaling, androconial scales intensively along those veins, all right? That's a characteristic of a lot of European frits, and it's also present in ours. This is a Callipi fritillary, and the male's upper left, female's lower right. It's not profound, maybe. If you got a 25 power lens out and looked at it, you would be able to see that the texture of those scales is different than the texture of just the scales lining the veins that are normal wing scales. And so it's worth, taking a 25 power lens, hand lens sometime and checking that out. But, we'll move along because there's a punchline coming, a really, really interesting punchline. So like these are skippers, and these are skippers are notorious for having pockets, evolved pockets in the wing that they took lots of androconia into. It's not quite sure how they move, the pockets, if they open them and close them. It's a lot likelier with the living wing that they're doing that. But this is also an example of the different androconia. And you see that some of the androconia are actually like spiculate, they're like little dots and little bars and stuff. You have to wonder if they're just not, dispensed, I'm trying to think of the deodorant packages that you see in men's rooms. Let's see, like little deodorant packages that are dispensed, and females are getting a brief whiff. It doesn't take long, but it better not, because you don't have long. So like here's Yuba skipper, and this is an example between a male and a female of the androconial pocket. the stigma is called. And there's a lot of different kinds of stigma, and the word itself, sex patch and stigma, are kind of exchangeable. I like stigma as being a pocket of scales, a sex patch as being a specialized portion of the wing that contains the scales, but it's not enclosed. You can actually almost see that these are enclosed. And if you look at the previous slide, you see that there's, that there are these pockets that they almost look like they're reversible. We never thought they might be. With living wings, they might be. So like, what's inside of those things? So this is a sex patch, and you can see the normal wing scales around it. Ordinary, one piled upon the other, not really looking too much different from each other. And then you see associated with the sex patch are two different types of scales. You've got these long filamentous scales, and then you have these more like I don't know what to call them, like shingle-like, plate-like scales. And that's not unusual, but they both contrast with the normal scales. They're specialized. Now, I can't even tell you what this is from because it didn't give it in the book. And this is an example of some hair straight stigma, the larger context on the left and the smaller on the right. And you can see that these patches can be, fairly obscure, but they're tucked away and they're obvious, and everyone who studied butterflies gets to know them pretty well. And these are, I borrowed this from the publication because it had a variety of scanning electron micrograph images, which I thought, would be, sort of useful to get a feel for these endoconia. And this is one of my favorites. This is an endoconial scale from Pieris. And if you think about it, it looks like, one of those bottles of dish soap you keep by your sink at the bottom. And then at the top, when you squeeze that dish soap, instead of squirting out at the end, you have these filamentous, applicators. I mean, it's almost like you can see what's happening here is that these ephemeral chemicals, these highly volatile chemicals are going up that neck and being dispensed out that filamentous section. And that's exactly what happens. What's interesting now in hindsight is that this is all happening within a living framework. These scales are not left to themselves after the butterfly ecloses. These sex patches are being supplied by wing hearts, by the butterfly blood. The whole system is operating as if they were, part of the body system. This is a live part of the butterfly. That means that, when a male goes to court a female and he gets rejected, he can regenerate. He can reproduce those chemicals so that the next time that he wants to court, and show his stuff, he will be able to do so. This is incredibly valuable, and it makes perfect sense in hindsight. One of the most important things that a butterfly does, beyond staying alive, which thermoregulation enables, is mating, and mate recognition has to be an incredibly important part of that activity. You don't recognize your mate, you're going to mismate. It's going to be just a mess, and life won't work. So in summation, I'm going to I worked on this a little bit. Sy et al. Have revolutionized how we think about butterfly wings and opened the door to thinking about sophisticated mating systems. Wings are alive. They are dynamic. They are instruments of fine-tuned temperature regulation. They facilitate the production of mate-locating chemistry well past eclosion. Maybe they are important after all. Now, see, people that don't know me don't know that I kind of downplay the importance of butterfly wings. That's sort of biology, behavior, biogeography. The wings, I mean, they lie. We know they lie. Monarchs and viceroys, butterflies that, will be cryptic, to blend in with the background, or, all of manners and ways that butterflies can deceive us. We know their wings are just not reliable, but they are important. And so this is startling. I don't think we've even begun to see where this is going to take us. I've talked with my colleague, Nick Grisham, and he says, I want to look at the genes behind this chemistry. it has to be important. You know, the genes are running the whole show, as we'll get to a minute. This is just like astounding. I was so happy. I mean, everyone that knows me or has spent time talking to me knows I'm so excited about this. And it's nice that you have pretty pictures. Just those are incredible things. And the concept of a wing heart, you know, that's really amazing and everything. But they're living. Butterfly wings are living parts of the butterfly. Very different from the way I was taught. Okay, so on to the next.
Okay, couple questions.
Okay, sure.
So this is all very, very new research, and so there may not be an answer to this right now, but do the pheromones come preloaded in on the males, like when they emerge, or are the pheromones synthesized in the wing, like in the scent gland, or someplace else and then moved to the, do we know that yet?
I don't know, absolutely no. I do know that the chemicals required for these pheromones are present in butterfly bodies. So like, it's not hard for me to imagine that a precursor is transported to the androconia, and it may be that exposure to air activates their evaporation. So I've heard some stuff and I've read some. I mean, there's an awful lot of stuff that you can read on the subject I haven't done yet, but that's a good question, and I want to know. I do believe that they're probably preloaded, or at least initial dose is preloaded. But it's interesting to note that a lot of those androconial scales are not deciduous. You know, there are some that are deciduous. In other words, they get cast away and they float about. But that Pyrus androconium we looked at, that is rooted, that stays there. So like, that's being fed and replenished. So what is replenished with is a legitimate question. I'll see if I can find out. It may be that nobody really knows, or it may be that one of those people, like I'm going to talk about next, does know, and I just didn't know about it, because you know what? I don't know everything.
Mm-hmm. So Bob Pyle wanted to know about the hair pencils on the tips of the abdomens of the Danaans.
What do they want? What does he want to know about them?
I don't know.
Well, the hair pencils and all of the abdominal androconial or sex attractant scales that's a whole nother ballpark. They're not on the wings. So like, I was focusing just on the wings. That's probably where I will find out about wing chemistry. You know, that's probably through that study that I will, unearth, information about, the chemistry of the wing androconia. So that's a legitimate question. I don't know. I don't know for sure, but.
Okay, and Gary suggested that cells may be carried along with the hemolymph through the wings, and so he's suggesting that there's possibility that pheromones are part of that.
Yeah. No,
yeah. So we can have more discussion.
Well, I mean, what it does is, I said, it opened up the door, and man, that's a big door. It's a lot of stuff. You know, mating is a very important thing. Proper mate choice is like essential. And so like, this is bottom line stuff. And I'm just like, yeah, I'm reveling in it, 2020. Yeah, that's recent. It's like,
yeah, exactly. It's very recent. Dave Nunley is asking if there's any evidence of sensory, any evidence of nerves or sensory organisms.
Well, they talk about sensilla. So yeah, and there is neurological network as to how well developed that is. And, like, that could be just involved with wing activities. It might not have anything to do with external sensing. But I would not be surprised, but what, like the costal wing, the costal margin of the forewing may have some nerve endings that are important for, for sense. I mean, you have to wonder what they were sensing, but I'm speculating. We do know there's a neural network. They said as much. So, what that means, to the butterflies and what they're doing is another matter. I don't know.
So there's a connection between the butterfly brain and the wing heart. Is that what you're saying?
Yeah, when we're talking about insect brains, we're talking about something a little different than, vertebrate brains. The wing nervous system is tied in with thoracic ganglia, which are sort of subbrains. And so like, there's probably not a lot of communication going on between ganglia regarding what goes on in the wing, so it's not like a vertebrate brain where we, send signals to various parts and we kind of have a coordinated all. But yeah, I would say that, talking about the ganglion or the ganglia, I think there's more than two in the thorax. It would probably have some feedback with the wing and make sure that, that needs are being supplied.
Okay, well, there's a comment from Gary saying that the entire wing surface is instrumented. So I've made a note of that. Maybe we can get back to that in the discussion afterwards, because I think there's a lot of interest in this idea
Oh yeah,
of wing parts and how they work, and the idea of living cells in the wings, which is pretty radical.
it is
So,
radical. Yeah.
yeah, so why don't you move on, and then we'll pick
Okay.
up that discussion after your.
Okay, so many of you know that Pelham is always talking about dissecting genitalia. I am not ashamed to admit it. We're going to have a brief discussion of a remarkable structure perched within the female genitalia, followed by some discussions about mating strategies. And it's going to be blue. I'm sorry, there's no way to get around it. However, I'm going to go to the next. I don't want to step on Nathan Morehouse. I don't want him to make me look like a fool when he does come and talk, because he knows more about this than simply anybody on the planet. So I'm going to be wallowing about in this field and demonstrating some aspects. And it's really important, in terms of the last presentation I gave, it was on Spirea coronis, and coronis. We talked about mating at or around mating sites, and then mating several times over the course of the year, and talking about what that was, because they don't lay eggs until the very end of that period. Why would they have to mate more than once? Well, now we have some answers, and we're going to look at that. So what I want to do, I'm not going to expect anyone to look at this and make any sense out of it. I'm going to read these things, and I'm going to do it in an interpretive fashion. Sex in a material world. Why the study of sexual reproduction and sex-specific traits should become more nutritionally explicit. I'm sorry, that is pornographic. I really think explicit and sex probably, needs to be, toned down a hair. But what they're talking about is sexual reproduction and sex-specific traits are the product of what the butterflies fed on as larvae. So it stands to reason that a female who had a very healthy nutritional history on a plant that was robust and good for it would be capable of supporting more eggs, maybe more healthy in general. And likewise, a male who had a good background nutritionally would be more fit and more subject to being selected by the female. That is basically what they're talking about here. I think it's important, to understand that this whole sex thing, with creatures, and butterflies in particular, dominates, their activities. I know we see them on mud and we see them, at flowers, and that's important. That's staying alive. But what they have to do is they have to find a mate. They have to mate. It has to be with the proper mate, and they need to find a place to lay eggs. So all of that takes a lot of energy and fitness. In a broadest sense, that healthy female mating with a healthy male and producing healthy offspring is the ultimate goal. And they're talking about this in this paper. Now this is 2010. 2015, they have I love this dynamic digestive physiology of a female reproductive organ in a polyandrous butterfly. Now, polyandrous means mating more than once. And that's what this is really all about. But it won't make sense until we look at the next one. Digestive organ in the female reproductive tract borrows genes from multiple organ systems to adopt critical functions. All right. The gene part is like, that's really abstruse, and it takes a little bit of I have to work through it with all that I got. But we're talking about digestive organs here. Digestive organs in the female. Well, basically, when I was talking about Spirea coronis, we were talking about, well, okay, so the female uses up all of the, the male sperm that was deposited, in earlier matings, and then needs to get mated again. Wait a minute, uses up. That wasn't, that nonsense. I mean, uses up. It had to dissolve it. It had to, incorporate it somehow. These folks are looking at how that happens. Okay, so the last one is, I don't know, I almost feel naughty. Structural complexity and molecular heterogeneity of a butterfly ejaculate reflect a complex history of selection. All right, so we're going to not focus on most of that, but the idea, the butterfly ejaculate, all right, this is males mating with females. They deposit their sperm inside of a thing called a spermatophore, which is a proteinaceous sac lunch in which the sperm are contained, and it is put into the female bursicopulatrix. And what they're talking about is the molecular heterogeneity of the butterfly ejaculate, what that means for the males that give it, and how females might be able to say, hey, this male is more appropriate or is better fit or what have you than another one. Now, I can't go to all those things because it's really like another whole field. And I don't want to get into, Dr. Morehouse's, wheelhouse, because, that's like a pitcher throwing, a ball right down the middle with the bases loaded. So, all right. So let's, go through some basic butterfly biology. Again, this is a male genitalia without the valve. And I always like to look at it as, you have this big hook on side and you got these two flaps, hook on top and two flaps on the side is kind of a triad, it's like, a threesome. You have two, these flaps that are basically, well, they're active. They actually are muscular and they, do participate in the act. The dorsal thing is like, it's really convoluted. It's pointy and it has spikes. Well, I'm inclined to think that those aren't accidental, and this has to do with securing the female. All right. Now, in this diagram, they give the intromittent organ. You like that intromittent organ? The name Ediogus, which is like, okay, I actually like to use the word Ediogus, at random and at lunchtime when we used to be able to get together for lunch, but it's equivalent to penis. And I wasn't going to do this, but I have to. We had a LEPSOC meeting in Park City, and a young lady from New Zealand gave a presentation on, weird genitalic evolution in micro lepidoptera, a certain genus in New Zealand. And it was like really interesting. It was fascinating. She did a great job of it, but she substituted the word penis for Ediogus and repeated it. I think I counted 14 times in her presentation. And she almost won the award because it was so entertaining. But I went up and talked to her afterwards, and she was beaming. And I thought, well, what? It's like, she said, I got to stand up in front of all these people and say penis as much as I wanted to. So I hope you're laughing. The female genitalia are all internal except for just the portion that copulates with the male. And this is important because this is where we're going. Now, as you see, there's kind of thing that's called the ostium bursae. That's the horny portion of the internal organs where the male intermits, and that's the copulation takes place through the ductus. The bursa itself is the whole thing, but the corpus is the sac there. Okay, and now you see these things called signa. It's important to see and remember the signa. These are internal things, and people have always wondered. I mean, they're very important taxonomically. When we actually do dissections, we note their shape and where they are, and so they have function for taxonomy. But we didn't really know what they were all about. We have an idea now. So I always show this when we talk about mating butterflies. I always show this because mating butterflies, it's not as passive as two spacecraft docking in orbit, but they're similar in that you have these equal and opposite flaps kind of coming together and joining. And anyone who's watched butterflies mate know that it's a lot more enthusiastic than this. But no, I really just wanted to show you my space photographs. But so this is the bursicopulatrix. It has the spermatophore internal to it, it's not really well documented. But if you look at that, what's called on the left of the pearly body, that pearly body is actually situated on the outside of the spermatophore. The round outside, the halo to the spermatophore is actually the bursa copulatrix itself. The spermatophore basically fills it. Okay, so you can see that the sperm are contained within the bursa. They are dispensed to the, I'm going to use some technical stuff, but they're dispensed to the egg by way of the, the ductus seminalis. All right. So like this totally separate system. The sperm's held in abeyance, and then it's dispensed as eggs are being laid. All right. So we basically have this idea: the males deposit, a spermatophore, and then this is used up. And as we have seen that they, if they get used up, then female gets mated again. But there's all a matter of cleanup. All right. And life is actually pretty tidy, even though we think of things that, you look at a carcass, right, and you let insects get at it and all the other things that want to get at it. It's not long before it's cleaned up pretty quick, right? And nature's like that, even though it might be messy along the way. All right. So now we're talking in the study, one of the studies done about the digestive organ. They were actually going right after the pieces, and it's like inside of the bursa, which is outlined there in black. Okay, there are these signa, and to the left there with the letter C, or to the right, the letter C is the signa. And what they call them, I like this too. The female signum in Pyrus rapi exhibits a chewing morphotype. Chewing, what are they chewing? They're chewing on these deposited spermatophores. They are eating the external part to expose them to this digestive organ in the bursa copulatrix, which then converts all of that to female energy. It was like, are you kidding me? I did not know this. I tried to find a picture of the organ itself, but it's kind of amorphous. I'm going to keep looking, and if I find something, I'll send you all an email. Then above that in B, you see actually the shape of the spermatophore. So this is like really useful. Now we know how, the butterflies that are polyandrous, that mate more than once, are able to deal with all of the detritus that's inside of them, because they digest it, and they have a highly specialized and very, very, very functional organ that is able to do that. So when Nate comes and talks to us, I'm sure he's going to have a bunch of things to talk about besides this. Maybe I can talk him into giving us a picture of that organ. All right. So some of you know that Pelham has been known to refer to males as useless bags of sperm. Don't let that Pelham guy tell you otherwise. Males are useful contributors to reproductive success. And I feel better personally knowing that. All right. I can't hear you laugh. I sure hope somebody's laughing at it. It's my jokes. These are the jokes, All right. So we're going to review classification and get into phylogeny. I hope I have enough time. I'm, maybe running a little bit over, but this is general stuff, and I don't expect anybody to have much to say. This is general stuff. This is circa 1961. If you look at this phylogenetic chart, because that's what this is a phylo tree. It's going to look like other things I show you. It hasn't really changed too much, except if you look at on the third branch down, what's called the Ethomyini and the Danaeini, those are actually in the same subfamily now. The same with Satyrini and Morphiny. They're in the same subfamily now. So there has been some changes, and that's kind of like where we're going here. We're going to look at, how things change, why they change, and like how to get used to that. And, I'll probably apologize because I'm responsible for some of the mess. All right. It's not a stretch to say that butterflies are, they don't have culture. They don't have tradition. They don't have history. They are gene driven, wonderful, amazing creatures, but they are automatons. They're molecular machines. I don't think that diminishes. I think that enhances our appreciation, because, you know, if we talk about organisms that learn, you know, we have the ability to put together personal histories and accrue information. Butterflies don't get that. They don't have a chance. They better do it right with what they got, which is, you know, pretty much, you know, what, you know, totally a genetic, you know, driven thing. Now there's epigenetic, that non-genetic, or they're genetic, but they are subject to being, you know, influenced by the external conditions, like, you know, photoperiod, you know, and temperature. So like, it's not like these genes are, you know, inflexible. But in a lot of ways, they are pretty inflexible. So when I began doing this, you know, we had nothing but the morphology, you know, which in butterflies, let's be honest. I mean, study butterflies because they're beautiful and their wings are beautiful. And wings are how we tell most of them apart most of the time. I guarantee you that it would be really hard for even the best butterfly guy to take a butterfly, you know, without wings and figure out what it is. He could if he had DNA, or if he was able to dissect their genitalia. I hope you're laughing at that one too. But at any rate, that was the state of the art when I began, you know. And I mean, you know, when Star Trek came along and they had the tricorder, I thought, wow, dude, we could just get the whole genome and with the press of a button. Well, it is upon us. Now, I don't want to assume that anybody knows everything that you need to know about DNA, and I don't want to bludgeon anybody with it either. But essentially what it boils down to is you've got four elements operating, four pieces operating, and they form, you know, nucleotides. Let's say the thymine and adenine always go together, and guanine and cytosine always go together, but they can be in any order. And, you know, it's like important to recognize that how many of these, you know, constitute, you know, a single gene. I mean, we're talking, you know, tens of thousands, hundreds of thousands. I don't know. I don't want to say millions, but I'll bet, you know, of nucleotides, you know, in an organism. So you're going to study genes. You're studying sequences of that look like I missed, like this. I mean, that's what basically a gene sequence looks like. And that is like, I know how exciting that is. I understand how everyone's going like, ooh. I mean, you know, if you had a chance to look at that or a morpho, I know where you're going. But so DNA, deoxyribonucleic acid, is a polymer. All right, which means it's, you know, a multiple, added to of long chains of nucleotides. You can read all that stuff. I don't want to really go into that too much because it's like I really get excited about it, but it's not the time in, you know, at this point in this conversation to, you know, go that way. But I did want to get this line in. Okay, DNA sequencing pulls the bases apart and then determines the position of each base. Yeah, it does seem like magic, but it's actually routine. And I was talking earlier, COVID has contributed greatly to general understanding of DNA, and people that like never before. They, you know, people saying, "Oh, DNA and, you know, sequencing and, you know, whatever." But so we just looked at the raw results. That's what, you know, basically it is. I know how exciting that is. So, you know, someday maybe I'll talk about, you know, the actual, you know, mechanics of converting those sequences into a phylotree. But I'm not going to do that right now because that's another whole thing. I think I might be in advanced butterfly biology. We might be a month or two away from that. All right. So at the end of all of the work that these researchers do, they have been put, you know, through the test. These trees are been trimmed and hammered, and, you know, hopefully they've done the best possible job they can to get to the, you know, the thing. All right. So basically every tree has at one end the twigs, which are the individual samples, and at the other end the trunk, which is the collective, you know, summation of how all the nucleotides all together are all unified at the trunk, and they only differentiate the farther away from the trunk you get. Now, that's kind of, you know, family trees. We all know how that works. So it's not really too bad. So this is an example. This is an example of one of the changes that one of my colleagues wants me to make, and I'm reticent. It's not that I don't really want to, and it's not that I don't understand what he's saying, but if you look at, you know, on the far left there, that little thing off to the left, that's the trunk. And you see a split, right? It comes and it bifurcates. One goes to the south, and it ends up at a butterfly called Symbrinthia brabira, okay? And then the other goes to everything else. And then so you go to the next, bifurcation, and one goes to Hypernarcia, levi, and the other goes to everything else. Well, in a sense, you could just take those out of the equation. They are basically, related, but really distantly related to the remainder. So the remainder includes all the ladies and all the tortoise shells. He shows quite clearly that the ladies are in one branch and the tortoise shells and angle wings are in the other. What he claims is they're equivalent, that the ladies are in Vanessa fine, but that all these others should be in Nymphalis. And I'm not comfortable with that. It's not necessary. If you leave them, as they are, you treat the angle commas as distinct genus, and you treat the Milbert's tortoiseshells as a distinct. It's like they're still in the same clump, but they're not lumped together, and people won't be confused. I mean, I would have a hard time coming to WAB and say, hey guys, all those angle wings that we talked about, Polygonia, they're going to be Nymphalis now. How you think about that? And they look at me and roll their eyes and say, well, common names are more stable, which in fact is, kind of the point. Oh, wait a minute. I have not accepted this. I have not adopted the conclusions. And Nick has spanked me several times and thinks that I should behave. But the remainder of the discussion that we're going to have here on the subject are going to be things that I have accepted. This is the tree of ringlets, okay? And like ringlets are ringlets, and people say ringlets, why are, ringlets are even a thing, right? But this is interesting because if you look at this tree, the trunk again is on the left, and it's got two major branches, right? And one branch includes all the things in the New World except the ones from Alaska and the Yukon. And the other branch has the Alaska, Yukon, and European sample. It's like, I actually had several references to this before this paper was published. They all indicated the same thing. This information was, well, let me read what I said. We are changing this. They were once Cynnympha tulia. They're now Cynnympha california, our ringlets. The Alaskan ones are still tulia. And this conclusion is based on more than this study. It was, Paulette liked this phrase, and so I'm going to say it. It was the camel that broke the brontosaurus back. In this case, Tulia in the far northwest is adjacent to, California, from just south of there, and the genetic distances remain. It's like they're different species. I'm sorry, not real sorry, but I'm sorry a little. And this is one that Cheryl Boleen was really interested in earlier. She was collecting or observing branded skippers. And these are all branded skippers. And she was observing branded skippers up at Slate Peak, and then at various elevations in the Okanagan Highlands, and by where she lives in the Chewuk Valley. And she was saying, "What's going on here? There's all these different things." And I said, "Yeah, it's a problem." But we at one time thought we had two species in our state: the common branded skipper in northern Cascades, and then the common branded skipper in the Olympic Mountains. This was traditional. I didn't make this decision. It was handed to me by people who were, more expert than I, that came before. And then everything else was, of another species, the Western branded skipper. And this study shows pretty clearly that that's just not so. You can't see. There's a paper that's going to be published later this month that will be definitive. Oops, I didn't want to do that. I'm trying to get this thing out of my way of my screen so that I can actually read. Okay, yeah, they don't have the skippers from Alaska that belong with the Europeans. So like the common branded skipper actually does occur in North America, in Alaska, adjacent to the Chukotka Peninsula. All the rest of ours are, the Western branded skipper. And you can see this tree, it's kind of like, right, okay. It's complicated. And the paper coming up again will address it. But, you know, it's like you can understand that when you see all these things down in the lower portion of this tree, they're all bunched together. Well, that's what you expect things to be really closely related to each other to look like. And you can see there's like little outliers, that, you know, little branches here and there. Well, a lot of times those represent, you know, geographic, you know, segregates that other people call subspecies that are, you know, not different enough to be different species, but they're different enough to be identifiable compared to other taxa within that. Now, this isn't definitive, and I'm just showing it as an example. When the paper comes out, I'll probably make notice of it, you know, because it's going to be, you know, involving people that have been in the Northwest, Andy Warren, and well, I guess Andy's the only one that's on that paper, but Nick Grish and my colleagues also, and it's interesting. And then you know, like this is like old news, but I thought I'd bring it up. Old news being 2019, isn't that something? Now, when you have these advanced, you know, techniques, advanced technology, what you find is that old is like anything beyond a year or two, right? So this is where we're at with that. And let's see, I think last but not least is we're talking about Fritz, and this is something I'm just gonna go there. Before 1947, everybody called all of our fritillaries Argynnis. Everybody did. It wasn't like a question. Spirea had been proposed by Scudder, but everyone said, now they just Fritz, and they throw them through Spirea in synonymy. All right. Then these guys, you know, really famous guys who did a wonderful job with fritillaries. I mean, their work stands as a monument to their incredible ability, and they're recognized to this day as being really remarkable people, and the job they did is a really remarkable accomplishment. But they got this one wrong, and they used Spirea and established the precedent, which took us to near the present. And there's a funny story. It's funny because it's not, I don't find it funny because I goofed up, but I advised James and Nunnally, David James and David Nunnally, when they put together their tome on the larvae for the Pacific Northwest. And I misspelled maybe it actually is appropriate, but I gave them my advice. And prior to publishing my catalog, I changed my mind, and but they didn't. And my golly, they were right. So they stand correct, and I stand corrected. Okay, so this recent paper was another one of those camel breaking back moments, and so like I want you to see this. This is like the lesser fritillaries are the Beloria there. You know, the lesser fritillaries are in that black portion of the tree. The variegated fritillaries are, you know, at the last thing, you know, the trunk of the tree splits into the variegated fritillaries and all the rest. And then the next split is between the Beloria and the Spirea and Argynnis. And it was just like, dang, man, we can't separate them. Like, they really are. Well, people had actually done some marvelous work showing that they were. And I was almost convinced I can blame Andy. Andy says, well, dude, you're going to have to explain to everyone, you know, Argynnis, you know, well, we have to now. I should have done it then, and then I wouldn't have to do it now, but I didn't then, and I'm doing it now. So this is, you know, our Fritz are Argynnis, and I'm pretty convinced of that. I'm pretty sure it'll stand the test of time. So now I get that names are, you know, an important part. You know, common names are less fragile than scientific names. I admit that. I mean, because of all the things that have happened in the last 5 years. But I'm doing my best to destroy that. You'll notice earlier on I used angle commas, right? Angle commas because some of those species have been called, you know, angle wings, and some have been called commas. And in the AKAs, like what, you know, Bob has done, you get both of them. So I said, hey, you know, I killed two birds with one stone. I call them angle commas and I get away. I made that up, and I'm allowed to do that. There's no rules governing that. There's people frowning and sending me nasty emojis, but hey, you know, I did that with green alphas, you know, green alphas. I mean, you know, we have hair streaks and we have elephants, you know, hair streaks have hair streaks and elephants don't. Green hair streaks don't have a hair streak. They're green elephants. And if you don't like that, you know, you can't hit me because I'm on Zoom. So at any rate, that is, the state of the game with scientific names. We have to make changes sometimes. They're uncomfortable. They're uncomfortable for me. You don't know what I have to do to revise the catalog for updates. It's massive. Sometimes the whole Guinness thing was a headache. I'm just getting it up to date now. I'm waiting for it to be updated. It's massive, difficulty for me. So it's not something I look forward to. I'm not torturing you guys, by changing scientific names. But the upside, you will not get arrested for using whatever names you choose. If you want to say Spirea, you can, it's fine. If you want to say Synonymphotulia, none of you guys would anyway. But if you did want to, you could. And furthermore, we'd all understand you. So I am at the end. Did I meet the time limit?
Just about. Gary, do you want to weigh in with your knowledge about the new book coming up?
John, while Gary's working on that, why don't you answer Lunabelle's question about comma and Colorado and BC?
What was the question?
Lunabelle, you want to give John your question?
Yep, I will.
Just do it out loud.
Hello. I was just wondering if you could give me some references to papers regarding the differences between the Western branded skippers and the common branded skippers, because in BC there's just a constant debate about which is which and what they look like. And
yeah, well, I just mentioned a paper that's about to come out, and I think Cheryl Bellin.
Yeah,
you
I
got
did see
that.
that. Thanks, Cheryl.
Yeah, that's one.
I just put it in the thing. The Kong et al. I just put on the chat.
Right. That is the most recent publication. But see, the problem is actually that they all kind of look alike, so it's going to take a lot more than just the paper to make a definition. The next one coming out will be a little bit better, but these are tough skippers. They've been tough for everybody for a long time, and I do understand the debate in British Columbia.
There just seems to be a real debate about Highland and Lowland branded skippers. I think there seems to be this, thing that Western branded skippers are the lowland species, while common branded skippers are the highland species. And it seems like every time somebody finds one in a place where they don't belong, then there's these assumptions of whether it's Oregonia or whether it's,
right.
And I recently ran into this because I found them in the location where I live where nobody has found them. And so some people think that they're Oregonia and some people think they're not Oregonia. And it would be great to have some clarification on that. And there isn't a lot of studies coming out of British Columbia about it. So.
Well, this is interesting. Actually, this is one of my favorite topics. I've actually written a little dissertation on the matter. If I can email you, I will give you the whole rundown. But I can tell you something right up front. You don't have the common branded skipper in British Columbia.
Oh.
They're all Colorado. That's by the criteria that have been established molecularly. Dave Nunnally would frown at me, and he probably is currently. Dave, are you frowning? Currently, the only places where the common branded skipper are in North America are far north Alaska, adjacent Chukotka Peninsula. Okay, so that does alleviate one of the problems. But for instance, the name Oregonia that you were using, that's restricted to Southwest Oregon. It doesn't occur anywhere in Washington or British Columbia. This is what happens when we don't know is we operate with a temporary name, a label name, and Oregonia became, I'm probably it's Bob's responsible for this. He was the one that told the state people that it's, it was Oregonia in the press.
Yeah, on your advice, man, on your advice.
I would rather blame Bob, but so we basically got the ball rolling the wrong direction. And so the name Oregonia doesn't apply either. honestly, there's one guy who knows the most about it. You probably know who he is. Crispin Guppy.
Yeah, I'm in communication with Crispin about it because he suspects that the ones that, so where I live is on Quadra Island, British
Oh, yeah.
Columbia. And the ones that I found here 2 years ago, he suspects are Colorado Oregonia. And I don't know. I'm just listening to the people that know more than me.
Well,
So
that's good. Well, given in the context of what I said about Oregonia, you're right. All the island stuff that we have, I mean, we have Patricia Morhutch, is online too. She's done a lot of those from San Juan Island, and these are the same kind of thing that you're getting in your island. Low elevation, maritime essentially. We get those all the way into Puget Trough. We called them all Oregonia. So you're calling them is in keeping with what we did call them. We call them the Salish segregate now.
okay.
Yeah.
Okay.
And that's just, amorphous. We don't have to put a name on it. We don't have a name to put on it to begin with, right?
John, you need to name that sucker, man. Just do it. Man up and do it.
Why?
Man up and name the butterfly.
I've manned up and not done it for a reason. I don't name butterflies unless it's by accident.
Another thing I just wanted to ask quickly was, I have found that there doesn't seem to be any, I don't actually ever know where to find them. Whenever I'm looking for any Hesperia, then I find them in random places. And it's not like a butterfly, like a moss elfin, for example, that's going to be around its food plant, right? Whenever I try to look for Hesperia, I just never know where I'm going to find them. And I've found them in swamps and I found them on top of tall bluffs, but there doesn't seem to ever be a constant place.
Actually, I think there is. I think if you look for the highest quality grasslands of many different kinds of grasslands, and what I mean by highest quality is native species of grasses. They have adapted to some exotic grasses, it's true. But in my experience, Colorado of all different types, the best place to look for them in general is the grasslands with the structure closest to whatever the native structure was there. That's certainly true on San Juan Island.
Well, the habitat that you talked about, the bluffs, I would say that open habitats are going to be necessary. First of all, bluffs seem like, balds on a bluff would seem like a likely candidate. And, Patricia can give you a bunch of information. She knows more about it than, anybody in the state. She's seen more than anybody in the state, right?
Okay.
Huh?
Okay, that would be great, because
yeah.
I'm definitely I'm really intrigued about it because it's sort of a random occurrence, and I'm definitely confused about the debate, and now I've just been calling them Hesperia because everybody seems to get uppity when I call them Oregonia.
do you get any other Hesperia up there? Or is it the only one?
This is the only one, but the thing is the debate between the common and the Western. I mean, people debate it so much that that there seems to be a problem in me calling it Oregonia, except for Crispin. Crispin seems to agree.
Well, we're on the same page. Yeah, Crispin is a good man to follow. He's an old friend of mine. I mean, he's old and he's a friend, so I guess
So Gary, are you able to join us now?
I think so.
Thank you.
Can you hear
Thanks
me?
very much, Lunabelle.
So, what question should we attack first?
You'll listen to you talk, man.
Oh, okay. Let me first say something about the wing being fully instrumented. Remember those pictures where there was a bright spot on the wing?
Right.
Well, that was a part of experiment where they're using a laser, infrared laser, to heat up locally a spot on the wing.
Oh.
They found that and they also measured as a function of time the heating up of that spot. And they found that there was a real critical temperature at which the butterfly would behave to get that out of the heat. They'd turn or so forth. And they did this in all different kinds of species. And the important thing is they did it on different parts of the same wing and they got the same response. And so this is why, as far as thermally, the entire wing is instrumented. But they also looked at sensilla you know, using scanning EMs and stuff.
Uh,
And over the entire wing, it's just peppered with these things. And then also there's neural structures connecting things. So yeah, they did a lot of cool stuff. And also they did field work. He went out, particularly with the white M hair streak, which is close to his home. And they looked at behavioral responses of the butterfly to wanting to heat up or cool off, depending on what they were doing. And then they also did some correlated experiments in the lab where they had a wide, now not just a point laser, but a broad heated source, and they present the butterfly to it in like a petri dish and then watch what it did.
yeah, I saw some of the petri dishes in the image.
And another thing just for all of you that are interested in learning more about this, the students did such great work, and in the supplementary data, there's all different kinds of video, like the pumping hearts in a bunch of different species and these behavioral experiments. So there's lots of video data that's accessible if you go to the website where you can get the paper, and then there's a click for the supplementary data, and
Yeah.
it's huge. And I've got all of that stuff on my computer. And so if anybody in particular is interested, I could send you or put it on Dropbox or something.
You can put it right on now if you wanted to. Share your screen if you got a movie ready to go.
Gary, I have a question for you about the motivation for the research. Was it, was the technique developed in advance and then it led to the discovery of the living cells, or is it?
History here. I, for 30 or 40 years, have been working with Professor Rüdiger Wiener at the University of Zurich on ants and bees and their vision, and he works on their behavior in the desert foraging. And I took my family to Europe in 2013. And when all the family went home, my wife and I went back and spent a week fooling around with having fun with Roger and Sybil Wiener. And I was working on polarization vision in butterflies, and I wanted him to give me some feedback on how he thought about my work, because he knows so much about that. And at the end of this, says, Gary, I have a request for you. He says, my the champion as far as the desert ant that can withstand the highest temperature is the Saharan silver desert ant. He says its body is covered with hairs. And if you look at it behaving in the desert, zipping across the sand, it looks like a ball of mercury. It's so bright. He says, so Gary, I want you to measure as a function of wavelength, the reflection from that hair coating it's got. And I told Roger, well, yeah, I can do that, but that's really not what you need to know. We have to do this in the infrared as well. And I know a guy, and this was Nanfang Yu at Columbia, who I met through Harvard. And he had just got an appointment there in the applied physics lab and was setting up a micro infrared lab. And let's see, I was talking to Roger like in August, and Nanfang said he should have his lab running by February. So I told Roger, well, I'll talk to Nanfang and see if he's interested. Of course, he'd be interested. So Roger sent me back with some desert ants, dead ones. And I talked to Nanfang and of course was interested. And so away we went. And the bottom line, we published a paper in Science, was what he learned from that is that this coating did two things. One obvious was it reflected the sun's visible light, but is what we didn't really understand until Nanfang and the students really worked on it, is that in the mid-infrared, where the core temperature was trying to get out of the body of the ant, it turns out that coating of hairs acted like a nanoreflection coating that made the emissivity like unity. So all that heat could get out into the cold, cold sky. So it was a way of dumping the core body heat into the sky. So it was a way of thermally controlling, you know, how hot they got inside. And of course, then the death temperature was much better because of that. So that was really an important insight. And then the wing thing started because then they were thinking about, oh, he got instrumentation like, you know, thermal cameras and stuff. And then they had, with NOMI, they were really interested in what was going on with these scent patches, you know, as far as like John was telling you about getting pheromones out to the female. And they were wondering, well, what about that as a structure that was had thermal control because you needed to, you know, get the molecules out of there. So that's what got them going on wings. And then they discovered all this other stuff. And that wing heart, the way that happened, I think they did it initially with Painted Lady. They stripped off all the scales on the wing because they wanted to know what they did as far as the thermal control. And they put it under the microscope and they noticed, you know, hemolymph going through the veins, carrying cells. And then they started, whoa, what's going on here? And they started looking into that. And then along with that, they discovered this pumping wing heart and got all excited. So it sort of evolved from that.
Gary, can I ask you how much you know or how much they were able to tell about communication of hemolymph between the wings and the body? John's right. For so many decades, we all said, oh, the wings are dead. It's just extraneous dead material with bonded membranes. Then you find that the unbonded parts of the membranes, as John said, have living tissue in them, and the hemolymph flows, which is, you know, so new to so many of us. But how much communication between the wings and the body?
They did something with this because they were wondering as far as the fluid being carried through the veins, what was that really doing as far as thermal control? And the answer was not very much. You know, it's really the surface area of the wings and how it dumped heat out. And as far as, I think they did something with connection of how does that fluid get to the wing in the first place? And I don't know the answer to that, but I'll find out. Because I think they did do something like that, asking sort of that question.
It was very cool for John to concentrate on this paper because it's really revolutionary.
Yeah.
Congratulations, Gary, on your part in it.
And I didn't even suggest it. John, I'm really, you made my day. That's really cool.
Hey, you made my year.
Yeah. And did you get that paper on the chrysalis that I sent
Yeah,
you?
I just did. Yeah. Yeah.
Because let me tell the group about that.
Right.
At Yale University, I worked with Kay Craig on spiders and spider silk and how they attract prey and stuff like that. And she ended up in Madagascar and has kind of, well, it's both moth silk and spider silk, and they make fabric and sell it and so forth. And Kay and I are good friends still. And so we were talking, I was telling her about this work. And then she was thinking, well, I have this one moth that makes a huge chrysalis, and it's up in the canopy, and it's fully illuminated by the sun. So how does it deal with thermal control? And I said, well, see, let's see if Nan Fang's interested in that. And man, was he ever interested in that, because the story is what that chrysalis is being wrapped with is silk that, if you look at look at a cross section of one of these silk fibers, it's got lots of little little holes in it, but they're all kind of random around. It turns out that the optics of what's going on because of the presence of these holes in the silk, it's a resonant structure in the infrared. It's called Anderson localization. And so that's what this paper is about. And so Nanfang again got all excited. And
So is it active temperature control in the chrysalis, or is it passive?
no, it gets rid of heat, the same sort of like the
Through
Yeah.
circulation of fluid?
No, it's passive.
No, it through two things. One, reflecting heat from the sun. And also if it gets hot inside, it can into the cold sky. Yeah, that's something you have to know. Oh, let me back up and say one thing. You get out of the shower and you're cold, right? Why are you cold? Two reasons. 50% is what you would think, just a cold evaporation of the water. But the other 50% is radiation. Actually, the heat in your body is radiating into the walls, the cooler walls of the shower and the ceiling. So its radiation is a really important mechanism. So like the desert ant and this chrysalis in Madagascar, the sky, the temperature of the sky is much lower. So as what happens is it radiates into the heat from the core is radiated into the sky. And as what you need is a coating on there that makes it really efficient as far as getting that heat through and up into the sky. And so that's what's going on in both the case of the ant and in the case of the chrysalis from Madagascar. Really cool. And all of this started because I wanted to visit Roger Vayner. I mean, just, it doesn't hurt to be lucky, John.
It doesn't hurt to be good.
Gary
Well-
also worked, speaking of Yale, Gary also worked with Charles Remington at Yale. Charles, people will know his name as the founder of the Lepidoptera Society. And Gary, that's when I first met Gary a long time ago. And Gary worked with Charles on the such things as the reflectivity and conductivity and convection and so on with the colors of butterflies such as the blue copper and the ruddy copper, right?
Yeah. That he was so good because, if I come across a butterfly and didn't know what it was, I'd get excited and go, and then Charles say, well, I'll take you up to West Rock and we can find some of these that are even better. And so at one, I worked with him for probably, I don't know, more than 10 years, 15 years. And I was finding all these differences in different species, could see different colors very differently. And I was at one point whining to Charles about, what does it all mean? What do I do with this knowledge? And he said, well, he had an idea. He says he knows a group of 4 sympatric coppers at RMBL, which he helped find, that where the males have different colors and they could mate, but they don't. And he says, if there's any place where reproductive isolation is visual, that's going to be it. So why don't we go there and work on that together? So we did. Yeah. That was just mad.
Can I ask Gary a question?
Yes,
Yeah.
it's open discussion. Please do.
Sure.
Gary, you mentioned that lady that went to Madagascar was studying the fibers that the moth and the spider had woven, and that they were hollow, and in the chrysalis they allowed the heat to get out, and they also made cloth out of it, you said. Now, was that cloth, if you make it into a shirt, would that shirt reflect the heat, radiate the heat into the sky?
That is such a good question. I don't know. She sells. In fact, I have a mask that I bought from her that's made from this stuff.
Well, put it on and see if you get cool when you go outside.
it's, oh, where is it? You know, I don't know.
Ah.
Yeah, so, I don't know that, I don't know how much of her, I can ask her, from that particular moth, what particular fabric does she make? Yeah, I don't know, but I can certainly find out. That's a really good
Awesome.
question.
If we go back to the question of the scent pads,
Yeah,
also known as sex patches, or
there are different things, the sense pad and the sense patches. Structurally
yes,
different.
well, they are. You're right, absolutely
Yeah.
right. But they're all composed of androconial cells, right?
Yeah.
And yeah, that's right. John mentioned that one's contained, one is more dispersed. And I remember Tom Lovejoy of World Wildlife Fund, he could never remember what the patch was called on monarchs, so he just called them hoosie patches. So some people just call the whole bunch of things hoosie patches. But the reason I asked John about the hair pencils on Danaeans, the monarchs and the relatives, there's an interesting convergence that goes on. John, I noticed in one of your pictures, one of the plates you used, that one of the cells had a developed ending with some fibers on it, and I believe it was labeled stink club, right? Stink something.
Well, first of all, that was from Jim Scott, right? So
Yeah.
that explains a lot
Well,
right
that
away.
explains the name, of course. Well, actually, he's quite right, because those specially developed androconia have a structure at the end that helps to disperse the pheromones even more. Well, that's convergent, sometimes in the very same animal, with another structure. It actually appears in the monarchs and the relatives, in the males. If you squeeze gently the end of a queen, or even a monarch, but it's more prominent in a queen or the butterflies of Asia called crows, they put out these great big brushes of fine hairs, two of them.
Hey, Bill Yake, squeeze the queen. It's time to make a poem.
Squeeze the queen, Bill, but write a poem about it. And these were called in German, that translates as hair pencils. They don't look at all like pencils in English. They're more like these beautiful brushes. But what they're doing is they're dispersing the pheromones. And so you'll watch queens flying along in the Everglades or someplace, and the males will be shaking their booties out there, and they're dispersing the pheromones in the air, and the females are flying along behind them, sniffing it up. Well, they don't get the pheromones from their abdomens as females do. They take those hair pencils, they bend their abdomen up to the wing, and they stroke their own
I'll be darned.
scent patches on the wings and saturate the hair pencils
Huh.
with the pheromones, and then they put them out and they shake them. So the stink club on the pheromone itself, I mean on the androconia scale itself, and the hair pencils on the abdomen, in a sense you could say they're convergently evolved organs to do the same thing. The main thing is just to get those pheromones out there so the females are smelling them.
That's cool. So it's just like a reservoir that is, yeah,
Yeah,
that's really cool.
we want that poem, Bill.
Yeah, we want that poem. Squeeze the queen, baby. Well, does anybody have more questions?
So John, yeah, I got one.
Oh,
So on the fritillaries, that other genus, I forget, the other European genus was on your chart that came out with our genus
yeah.
on top. What is it? What's that other?
Fabriciana.
Yeah, Fabriciana. So
Yeah.
that's being sunk into our genus too, right?
Exactly.
Yeah.
Gamora, Sagana, all of them are being sunk. It's all one big genus with subgenera.
I think it's pretty cool that Nunnally and James got it right, way ahead of the game.
Yeah, I think it's pretty cool too. I'm just sorry that it was at my expense.
Well, that's only part of the story. The whole story is there was way too much trouble to change it at that late date.
But it makes sense. If you look at a high green or a high brown fritillary or a dark green fritillary, as they call them, say up in the Lake District of England, and then you look at an Atlantis or or a Zerini fritillary here, you can't imagine that they're really in different genera. It just doesn't make any sense.
Yeah, I feel the same way about viceroys and lorquins.
Well, they're in the same genus.
Well, I'm saying they're just as different.
No, I'm saying that these fritillaries in England, they look exactly like American frill.
Oh, yeah. Well, I mean, how about Childrina? I mean,
Well, and yeah, and the Paphia, the silver wash fritillary,
right.
they look quite different. Yeah.
I think that the problem that we have is that we get accustomed, and as soon as we're accustomed, then we become presupposed, and presuppositions lead to assumptions, and the next thing you know, we're wrong.
Well put. Great job, Johnny.
Well,
Yeah.
thank you very much. Look,
Thank you.
it was my pleasure. You just don't know. I missed you laughing at my jokes.
We were laughing, John. You just couldn't hear it.
There you go. So thank you very much, and make sure that we get a hold of. Is it Maribel?
Lunabelle.
Lunabelle, yeah. Make sure that we get I want to give her the best opportunity to do so. I think she signed off, right?
I don't know if she did or not. I'm not sure.
All right. Well, I need to give her the information she so desires, because she does seem to want it,
So Cheryl posted a reference that you referred to, and so I don't know if there was something more that you were suggesting.
Yeah, there's the current. Well, there's two things. One is my own concoction, which of course is full of words, describing the situation in the Pacific Northwest. It would help her a lot understand like the names and the non-names. Well, the thing that Cheryl posted was a published paper that apparently I asked her, and she saw that and got it. I think she got it down. If not, she will. So the only important.
Carolyn may be able to provide you with Lunabelle's email.
Okay.
Carolyn, is that true?
Yes, I was planning to do that.
All right.
Of course
Excellent.
you were. Of course you were.
Got it.
Okay, well, thank you so much, John. This has been really, really interesting, and it's so wonderful to hear about such recent groundbreaking research. And so we're really thrilled that you were able to present it to us.
Well, you know what I'm thrilled about? I'm thrilled that Gary came on and got his just reward.
Thank you, Gary.
John and Bob, are you still there?
Yeah, I'm still here, sort of.
Yeah.
I've been working with a woman in Sweden, and we have a really, I think it's going to be a really important paper that has been accepted by the proceedings of the National Academy of Sciences.
Well done.
It's now sending back the final proof, so it should be released next week, beyond embargo, so I can share it. But I'm really interested in seeing what you guys think. It's like coevolution. It's the thing about red sensitivity evolving, but along with it comes what's going on with the two of the other blue and green photoreceptors that come along for the ride to give you better color vision in the green for foliage, but also in the red. I mean, and it's with the Atala fritillary, Eumaeus Atala, and then the Japanese oak blue.
Oh.
Those two species. And it's this Marjorie, she is just amazing, but it gave me an excuse to publish some of my work, on the visual pigments. So I'm really interested in knowing what you guys think of this. I think it's really, really an important, her work is so good.
Well,
That's great, Gary, but it's all just space lasers.
Gary wanted us to know that these two papers have now been published, and that the PNAS paper shows that evolution tunes spectral properties of butterfly vision to the spectral properties of butterfly wings and food plant. If you want to copy the citations, you can pause the video. Here is the first publication, And the second.
I was wondering.
John, I can't resist asking this question. We've all been thinking about it and everybody's been afraid to ask it. But we've all seen rags, butterflies with their wings half worn away.
Hmm.
Are they flying around in excruciating pain?
Absolutely. I mean,
Yeah.
like,
What about pain in butterflies?
pain is a wasted resource. I mean, if you have nerves that are devoted to pain, then you're, like saying essentially you can do something about it. If a butterfly gets picked out of the air, experiencing pain is a waste of, of nervous tissue. I don't want to say, they don't feel pain because they try to escape, but when it comes
Well,
to
they do
wings,
just fine with bird marks on their wings, don't
yeah,
they? Crunch.
yeah. Yeah, so like that, I think the bird strikes on wings indicate that they're not feeling pain. I would be more concerned about whether they leak. Like if you have a strike in a wing, are you going to link hemolymph? And I'm wondering if there is a way to heal that. You see what I said? It opened up the door to all kinds of things. We have just begun to plumb where
Isn't
this
this
goes.
fun?
Huh?
I mean, that's a really big question. I concur that I doubt very much that there's pain associated with the wings. I don't know about the body, who could say, but the way in which they behave when they're highly damaged. I mean, David Myers, are you still here? David has his wonderful, brilliant, gorgeous butterfly book that I hope you all know called Wings in the Light, published by Yale University Press. If you don't know it.
I don't know it. Wings in the Light?
David, please get together with Paulette or Carolyn. Carolyn, perhaps you could put it out on the members.
There you go.
Yeah.
About David's
Look at
because,
Melissa and Patricia's website. They got it up, showing it now.
oh, good. It's one of the most beautiful butterfly books there is. But one of the things that distinguishes David Myers, who used to be my fellow Wakayakum butterflyer down here, until he moved down to the part of the world where Nabokov said he went into an absolute butterfly mania down in Ashland, is that David, as a butterfly photographer, does not dismiss or eschew the regs, as you call them, David, the old collector's term, the butterflies that are badly damaged. David pays special attention to the butterflies that have had a history. And it's very good because it shows that these butterflies have lived and they've had experience, they've had a life, and you can conjure on that. They're wonderful, wonderful photographs. Almost nobody else. Oh, I mean, Cheryl and Carolyn and Melissa, Melanie, and others do photograph these butterflies, but David makes a specialty of it, and they're absolutely wonderful. So you look at these butterflies that are just, I mean, less than shadows of themselves, and they function very well. I remember an Edwards fritillary, great big fritillary from Colorado, the only fritillary in the country that has no subspecies, I think, except for Diana. And it's a big butterfly, 3 inches across fully. This one couldn't have been an inch and a half. I mean, it was less than 50% of its wing length. Joel Kingsolver, who used to be at the University of Washington, did research, Gary, you probably were involved with him, that suggested that butterfly wings were evolved for photoradiation or warming up to about a third of the way out. And after that, flight took over. But you can fly after you have about a third of the wings. This butterfly had no more than a third of the wings. And it was behaving, it was trying to mate, it was gliding, it was doing everything else an Edwards fritillary does practically, but nobody was going to collect it, so it was safe, you see. So I don't think they feel pain, Dave. And I don't know about leakage, Don. It's a darn good question, but there's so much more to learn.
They just seal off.
Yeah, maybe so.
Hey, Melissa, Patricia, hi. Good to see you guys.
So, Bob, thank you for those kind comments and good description of the book. I am especially interested in photographing rags and things. And for the most part, they're old butterflies, which have a lot of wear and tear, just like I myself do. But I did find something really interesting this last year of a quite young Icaricia, probably, I forget whether it was Ackman or I think Lupini, but very young, that had a whole lot of missing fringes all the edges of the wings had been beaten up, and yet there were still all the very fine, delicate, long hair-like scales on the body and on the wings near the body, which made me think it was young. So accidents happen to young ones too. The book is Wings in the Light, Wild Butterflies in North America.
I'll find it.
It's
Thank you.
wonderful.
Yeah, my butterfly book doesn't have any pictures.
Yeah, but
Hey,
it's perhaps the most important one we have, John.
Can I ask you three a question? Do you see any correlation between durability of a butterfly species and its size?
What do you mean by durability?
Well,
Longevity.
No, this is a loaded question. The Western pygmy blue, that thing is so bulletproof, I can't even believe it. I worked on a single male for a week in my feeding it every day, but you know, it just, I couldn't believe such a very tiny butterfly, how incredibly durable it is.
Oh,
I think it's a mass thing. The bigger you are, the harder you fall. When you're tiny like a pygmy blue, you just get blown around. It doesn't matter.
Nobody notices you. You're so tiny.
Right.
well, we do think of swallowtails and monarchs living longer than a lot of other butterflies. Of course, monarchs have a reason. And then the long-lived nymphalis, which we still get to call nymphalis, as differentiated from the Pelagonians. Thank you, John, for that gift. With their big, thick bodies, they're highly muscularized thoraces. They live a long time. Of course, they overwinter. They're adapted for it.
Yeah.
But I don't know. That's a really good question about size and longevity and durability. Gary, I don't know. I think it's more structure.
Yeah. Anyhow, it was a surprise. I'm in admiration of that butterfly.
That's a good story.
By the way, I'm working with Adriana Briscoe on that one. So it's really an interesting butterfly.
Yeah, you keep
I've been
good
looking
company.
and looking for more of them in Washington. I did very little in the field this summer, but one thing I did was spend a heck of a lot of time in over in Benton and some of the counties around there looking at so much Russian thistle and rabbit brush, trying to find another pygmy blue. You know, I found the only one in the state, and it's still one. And there got to be more. So you people get out there and find us some more pygmy blues. They got to be out there.
I can't believe Adriana Briscoe, she has them on campus, a big population just right on the UC Irvine campus.
Oh, yeah.
Down in the, you know, in the gushy part. It's amazing. Anyhow, it's pretty nice for us.
Well, they feed on tumbleweeds, you know, like there's not much they can't eat.
Just so long as it's a kinopod. And if you find those Russian thistles right next to rabbit brush, it's the perfect location for them.
Oh, I'll remember that. I'll write that one down.
It's not a thistle. Russian thistle is a, you know,
Tumbleweed.
The big
yeah, it's
one?
tumbleweed. It's tumbleweed is what it is.
Okay. Great, thank you.
I think the pygmy blues have quite a few generations too. I think they just keep pumping generations out, maybe 5 a year.
They sure could, yeah.
How quickly do they go through?
Pretty fast. So I've only reared them once, but I think I took them through from egg to pupa in a week.
Wow.
Wow. A week?
Yeah.
They're so tiny, I guess there's not much to do, huh?
The reason I know that is we were actually staying down in 29 Palms in California, and the things were flying around the bushes outside, and I collected a female. She laid the eggs, and we were only there about a week, and I was already getting pupae before I went home.
Fantastic.
That's amazing. They're also extremely durable. On the butterfly big ear, I was in the Southwest too early. It was too cold that spring of 2008. So I never found the Sonora blue and the orange tip down there and things I was looking for. But in Death Valley, it was late February of 2008. And there were a lot of flowers already, but it was just too cold. There was nothing flying yet, except for one butterfly. What was it? A pygmy blue, of course.
Wow.
You can find them abundant in of all places, Honolulu, right behind Waikiki Beach. There are some swamps and canals. Pygmy blue all over the Salicornia there.
Oh, Bob, that's really good to know. My granddaughter has just been accepted at University of Hawaii at Manoa.
Yeah.
This fall. So I'll definitely be visiting her.
And there's another cool introduced blue there. What's called the grass blue? What's the name of it, John? Zizula?
Zizula, yeah.
And that was found just a couple of years before Tia and I were there in the big year, and we were
Oh,
there.
no, Zizeria. Yeah, you're talking about Hawaiian thing.
Yeah, Zizeria. And it was only found a couple of years before that on the Big Island and on Honolulu, on Oahu. Tia and I were there in December of 2008. And it was all around the back of Waikiki, the grasslands behind the beach too, that and the pygmy blue. We found the first ones on Kauai. Tia did.
That's
Okay, well, we're
thank you
running out
for
of
that
time.
one. That's good. I'm sorry.
We're getting, I think we're probably at the end of our time for Zoom. I only scheduled for 2 hours, but this has been an extremely interesting meeting. So thank you very much for joining us, Gary. We really appreciate it.
Oh, you're welcome.
Thank you, John, for your presentation and for everybody else's input.
So good to see all you.
Okay, we'll see you next month. All righty, stay safe.
Yeah, see ya. Bye.
Bye-bye.
Good
Thanks,
job,
everyone.
John. Thank you so much.
Bye, Bob.
Raina.
Oh, Raina, how you doing? Hey, how's that
Good to see you,
kid of
Brad.
yours?
Good to see Twig at a meeting.
All the kids have flown the nest.
Yeah.
Raina and Jay, so good to see you.
And
Good to see
Mariah
you.
Robinson is butterflying in Europe. She got her doctorate.
Yeah, I heard a little bit about that. Yeah.
She's having a great career, yeah.
Yeah.
Bill Yake has a wonderful new book out too. It's an absolutely gorgeous book of poetry. Tell me the name again, Bill.
Squeeze the Queen.
Squeeze the queen. We'll look for that.
There, okay, Waymaker.
Waymaking by Moonlight.
Waymaking by Moonlight. Absolutely beautiful new book of substantial book of Bill's collected in new poems.
Ah, beautiful.
Adios. Adios, amigo.
Adios, everybody.
Right.
Bye-bye. Thanks again.